Hello everyone. This will be a slightly different video or stream or whatever. Usually I stream all my lab stuff that I do, but yesterday I didn't really have time to do that. I I actually have a little bit of a time crunch because I'll be attending the Northeast Algae Symposium, and I have a deadline of the 16th to submit an abstract for a poster, and I still need some data before I can even enough data to be able to actually put a abstract together, and I'll probably be getting that data a day before the deadline. So yeah, yesterday what I was doing was I was actually calibrating this thing. It's a thing to count algae cells essentially. You put the vial in there with a sample, and it spits out a number, which is the voltage which you convert to current, and then you can, by plugging it into an equation, you get the density. And the way it works is, as the light goes through, some of the light is absorbed by the algae, and it's a specific wavelength. I believe it's 540 or no, 590, I believe. But it's a specific LED light in there that gives off a specific wavelength, and that's absorbed by the algae. So what I was doing was I was calibrating it by what I took a known sample size of algae or density. I counted it using the hemocytometer, and then in doing so, I was able to then make a serial dilution, which is essentially, I did a 10% dilution, so it went from 100%, 90%, all the way down to 10%, and then I did 5% just to get extra precision towards the end. And the idea, if you do it right, is it should show a linear graph. So like 600,000 cells, 500,000, and so on. It should show a good linear graph. And I'm actually going to show you the graph that I was able to get. Okay, now this is the graph that I got. This is actually an Excel spreadsheet that I use, almost like a graph sheet or spreadsheet. Like, I do all sorts of stuff in here just as to calculate, to make things that I just need to print out but I don't actually need to save, or to just mess around, and then eventually I transfer the data to an actual spreadsheet. This is the graph that I was able to come up with the calibration curve, even though it's not really a curve, but it was able to get I believe the blue line is the expected, and the orange line is the actual amount. Now I didn't scale it properly, so that's why you see like a little I guess hiccup towards the end, because it goes from 20%, 10% to 5%, when really it should be scaled down a little bit because the interval isn't the same. So it looks a little different, looks a little stretched out. But that's just a preliminary graph that I just messed around with. I'll eventually transfer it to and scale it properly. It's pretty spot on to what it should be. So essentially what you do is you then you get the absorbance factor, the what is it called, OD, optical density, and then you're able to get that and then correlate it to to the known count, and then you come up with an equation, which this is actually, let's see if I can show you the equation here, right there. Essentially, it's the log of 400 divided by V, which is the voltage that comes out, times 2,083,535 equals the trichome count. Now, the count itself, I can only get to 3 significant figures, because that's how much the voltage is. The voltage reading is on here. It's because the top voltage is 400 millivolts. So, and it's 3 significant figures. So obviously at the end you can only be 3 significant figures in accuracy. But that gives me more than enough accuracy for what I'm looking for in terms of error bars. It gets me within significance that I need. And let's see. Now I just gotta deal with all this stuff here. But so I was doing all this until, I don't know, 10 o'clock last night. Well, I was doing the actual stuff, the calibration part, and then I have to do all the calculating, which I did using that spreadsheet and so on. Which now it's funny because I was lucky because my timer is off by an hour now that the daylight savings time thing is around. So I was able to stay up till 10 in here with light, but at 10 o'clock all the lights shut off to not mess with the algae. And so I was sitting here calculating in the dark for another hour or so on the computer, and then I moved out into the kitchen to do all my notebook work. So that was my night. My goal is to be able to get some enough data by the 16th to be able to actually put together an abstract to present at the Northeast Algae Symposium. And not present an abstract, but because I'm actually going to be doing a poster. The hope is that I actually have enough data to make a poster. But so I'm cutting it close here, so that I'm doing some I guess all-nighters to be able to get. Now I'm just waiting for a. I'm actually upgrading this incubator. The incubator will stay the same, the size and everything, but right now the grow light that I have in there is like maybe 10 centimeters by 10 centimeters, something like that, and there's not enough space to put these and these on there. And because there's such a time crunch, I need to have a large enough sample size to be able to have enough significance to report. Like I'm going to continue to perform the experiment again after the abstract is submitted to get some more refined data, but I need to at least have enough data to report. But to do so, I need to have a large enough sample size, which this is a large enough sample size, but I don't have a large enough sample or not large enough light. So what I'm going to do is I'm actually going to have the entire incubator will have lighting. That way I don't, because one of the issues is it's not just the fact that I have a small light. It's the fact that it's the thing that holds up the light, the frame, takes up a lot of space. And not really a lot of space because it's only like, two little, leg areas like that. But like it's in the way. So if I can remove that and put the light on top, like a large light that will cover the entire thing, I can put my samples anywhere. And not only that, there's another issue of the aeration lines, that because there's this light right in the middle, and to disperse the lines everywhere, the lines are on top and then they have to go around. So you have this issue of having to move around the samples but without running into the light. So by eliminating the light completely inside of there, I should be able to have a heck of a lot more space to move around things. Anyways, I gotta label this. Yeah, pretty much as soon as I was done with everything, I pretty much closed up what needed to be closed up, and then I just left everything where it was, dirty and everything, because I needed to get the stuff done in here before the lights turned off, get all that stuff done. So, and then by then it was like, I don't know, I think I was done at like 1 o'clock. Yeah, so I'm waiting today. Later today, the lights should come in somewhere around noon. I'll start setting up this experiment and putting it in there, and hopefully by the time I'm done with this, the lights should be in, and because they are on their way, and then I'll be able to install the lights. That's it for right now. I'll get back with you when with my next thing, I guess. Thanks for tuning into this little, I guess, cleanup.